Leaf. Leaf is a tiny object in the universe. But leaf is not as simple as we think. It seems to be exaggerated if I claim that leaf is a large factory that produces glucose and oxygen, which are most essential substances for the life on the earth. Let us travel deep into the leaf and study the reactions that takes place, which are called as photosynthesis.
Green plants prepare their own food by photosynthesis. Hence, they are known as autotrophs. Photosynthesis is a physiochemical process in which light is used to prepare food. Hence, it is evident that photosynthesis is the basis of food for all the life on earth. And it is the process that releases oxygen to the earth. We already know that chlorophyll, light, carbon dioxide, and water are necessary for photosynthesis. Joseph Priestley, who discovered oxygen, identified the role of air in the growth of green plants. He used a bell jar and a candle in his experiment. He lit the candle and covered it with a bell jar. After some time, he observed that the candle got extinguished. He then performed a similar experiment using a mouse and a candle, and observed that after some time the mouse suffocated to death. Priestley repeated the experiment, but this time he also placed a mint plant along with the mouse inside the bell jar. This time he observed that the mouse stayed alive and the candle burned long after he expected it to. So he concluded that the plant was able to replace the air that the mouse and the candle have used up. Another scientist, Ian Ingenhouse, used the same setup as Priestley. To understand the importance of sunlight to the plants, he conducted the same experiments in the dark and in sunlight. He also performed this experiment with aquatic plants in bright sunlight and noticed that the small bubbles released by green parts of the plant were oxygen. He demonstrated that only plants can release oxygen. The energy for photosynthesis originates in the sun and arrives at the earth as sunlight. The light has both a wave and a particle nature. The particles or photons are the smallest units of light. Photons oscillate along a path which is measured as a wavelength. The light emitted from the sun contains photons in a wide spectrum of wavelengths called the electromagnetic spectrum. Photosynthetic organisms use only a small portion of the electromagnetic spectrum called the visible light. Photosynthetic organisms contain pigments that facilitate the capture of wavelengths of light in the visible light range. The color of the pigment comes from the wavelengths of light reflected. Plants appear green because they reflect yellow and green wavelengths of light. Red and blue wavelengths of light are absorbed by these pigments and provide the energy that is used for photosynthesis within eukaryotic photosynthetic organisms, also known as photoautotrophs. The chemical reactions of photosynthesis occur within plant cells in specialized structures known as chloroplast. Photosynthesis consists of two sets of reactions, the light-dependent reactions and the Calvin cycle. Within the chloroplast are small disc-like structures called thylakoids. These thylakoids are surrounded by a fluid-filled space called the stroma. The reactions that synthesize the glucose, the Calvin cycle, occurs in the stroma. The light-dependent reactions occur in the thylakoids. It is here that conversion of light energy to chemical energy is initiated. In most photosynthetic organisms, thylakoids contain pairs of photosystems called photosystem 1 and photosystem 2. These photosystems work in tandem to produce the energy that later will be used in the stroma to manufacture sugars. The photosystems of the thylakoids consist of network of accessory pigment molecules and chlorophyll, the molecules that absorb the photons of light. Within the pigment molecules, they absorb light energy, excites electrons to a higher state. Photosystems will channel the excitation energy gathered by the pigment molecules to a reaction center chlorophyll molecule, which will then pass the electrons to a series of protein located on the thylakoid membrane. Photons of light strike photosystems 1 and 2 simultaneously. We will examine what happens with the photons striking photosystem 2 first. The energized electrons are passed from the reaction center of photosystem 2 to an electron transport chain. The electrons lost by photosystem 2 are replaced by a process called photolysis, which involves the oxidation of water molecule producing free electrons and oxygen gas. While this oxygen gas is a byproduct of photosynthesis, it is an important input to this cellular respiration pathways. As electrons pass through the electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to the thylakoids, creating a concentration gradient. The gradient powers a protein called ATP synthase, which phosphorylates ADP to form ATP. The low energy electrons leaving photosystem 2 are shuttled to photosystem 1. Within photosystem 1, low energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the electron carrier NADP plus to NADPH. When the chloroplast is receiving a steady supply of photons, NADPH and ATP molecules are rapidly being provided to the metabolic pathways in the stroma. Therefore, the ATP and NADPH formed during the light-dependent reactions are used in the stroma to fuel the Calvin cycle reaction. Calvin, James Bossam, and Andrew Benson worked out all the biochemical reactions of the C3 cycle. C3 pathway was named after Melvin Calvin as the Calvin cycle. It has 3 stages: carboxylation, reduction, and regeneration. In carboxylation, the 5-carbon molecule ribulose 1,5-bisphosphate accepts a carbon dioxide molecule to form intermediate 6-carbon molecules, which then split to form 2 carbon molecules of 3-phosphoglycerate. This reaction is catalyzed by RUBP carboxylase. Ribulose bisphosphate carboxylase also exhibits oxygenation activity. Hence, it's called ribulose bisphosphate carboxylase oxygenase, or simply Rubisco. This phase is called carboxylation because a CO2 molecule is added. Reduction stage, a series of reactions leads to the formation of triose phosphates, which act as precursors to glucose and other sugars. At first, 2 molecules of 3-phosphoglycerate is phosphorylated to 2 molecules of 1,3-bisphosphoglycerate by utilizing 2 molecules of ATP in the presence of the enzyme phosphoglycerate kinase. Second, the enzyme glyceraldehyde 3-phosphate dehydrogenase reduces 2 molecules of 1,3-bisphosphoglycerate to 2 molecules of glyceraldehyde 3-phosphate. 2 molecules of NADPH are used in this reaction. Thus, to fix 1 molecule of carbon dioxide, 2 molecules of glyceraldehyde 3-phosphate, 2 ATP and 2 NADPH are utilized. 6 carbon dioxide molecules are necessary for formation of 1 glucose molecule. Hence, let's calculate for 6 carbon dioxide molecules. 1 carbon dioxide molecule results in 2 molecules of 2-glyceraldehyde 3-phosphate molecule. Hence, 6 molecules would yield 12 molecules of glyceraldehyde 3-phosphate. Of these 12 molecules, 2 of them are used to make 1 molecule of glucose. The remaining 10 glyceraldehyde 3-phosphate molecules make up a total of 30 carbon atoms. These are involved in the rearrangement of 6 molecules of ribulose bisphosphate, a 5-carbon compound, thus balancing the reaction. In the regeneration phase, 1 ATP molecule is utilized to form 1 RIBP. Hence, during the Calvin cycle, 1 carbon dioxide molecule needs 3 ATP molecules and 2 NADPH molecules for fixation. Therefore, the net utilization of energy for formation of 1 glucose molecule would be 18 ATP and 12 NADPH molecules. So students, make sure not to forget the importance of plants in our lives, or we will likely be ending up like the mouse in the bell jar.